Hey, one of you requested that I explain the patterns of inheritance myself, so I'm gonna try my best. But before I do that, I really need to have a reassurance that you still retain and you still understand what genes and chromosomes are. So here we go. Now, a gene is basically a sequence of nucleotides in our DNA that code for a certain trait or different traits. For example, if you're looking at this picture right here, the United Color of Benetton, um, we're looking at distinct traits that were genetically passed down, and the same traits are also, are also inheritable to the next generations. So, for example, we were talking about traits. So, traits could be distinct or um, non-distinct, or they could be are not as easily distinct, or they could be not visible at all. But because we're looking at visible traits right here, so the best example, the one I can figure out would be maybe hair texture, as well as hair color and also skin color. So these are traits that have these people have inherited from the parents and can also pass them down. If you look at a simpler organism, for example, Mendel's pea plants, we look at traits like flower color, pink in this case, and then yellow in this case. Also pod shape, smooth and undulated. In either case, both of these organisms are diploid organisms, meaning their genetic makeup is the result of two parents, or their genetic makeup is a combination of two individuals. Um, Here's a list of some very important vocabularies that we need to understand and be able to use when we deal with genetics and the patterns of inheritance. The first of which is homologous chromosomes. Homologous chromosomes, and let me just go back to the picture for a second. So basically, we get our information from two sets of parents, and they come in the form of chromosomes. So if you look at this picture right here, the yellow chromosome over here was inherited from the mother, and the purple one over here is the paternal chromosome, or something that was passed down from the father. Now, they are not genetically identical, meaning they do not have the same genetic information, but they are identical in terms of their length, size, as well, and they look at the positions of their centromeres, as well as their gene loci. For example, if this is a banding, or I mean, if this would appear as a banding in a karyotype, but if this is the locus for, say, the eye color gene on the mother's chromosome, on the same locus would be the eye color gene on the father's chromosome. With the and this applies to other genes. For example, the hair color gene locus would be the same on the maternal and paternal chromosomes as well. Um, Now, an allele is basically variations of the same gene. They usually, for this unit, we're going to be looking at two versions of the same gene, but there can be more, and we're going to look at more variations than two as we go along further in the unit. So, for example, if an individual has eye color gene that is blue and an eye color gene that is brown, they have two alleles for that. I mean, they do have two alleles for that, but in this case, both the alleles are different, meaning they are homozygous for the trait. Sorry, they're heterozygous for the trait, meaning both alleles are different. But if they were to have blue eye and blue eye from both parents, then they would be homozygous for that trait. If they were heterozygous for the trait, meaning if they had a blue eye gene, and then they had a brown eye gene, but the person appeared to be blue-eyed, then the blue eye gene would be dominant to the brown eye, and the brown eye would be recessive to the dominant gene, which in this case was assumed to be blue eye. Now, the dominant gene is the one that gets, the dominant allele is the one that gets expressed, even though the recessive allele is carried, it is not expressed, meaning it doesn't show physically. The one that shows physically is the phenotype, determines the phenotype or the physical appearance of an individual. The genotype is the genetic information carried within that individual. So going back, if this person was heterozygous for eye color, meaning they had a blue eye, a gene for blue eye, and they had a gene for brown eye, then they would be heterozygous for that, but phenotypically they would be blue-eyed, but genotypically they would be heterozygous, meaning they would have two different genes for the same trait. Now, one more thing we need to talk about is independent assortment. And independent assortment is basically the random sorting of chromosomes during meiosis. Now, we remember that diploid organisms have a pair of homologous chromosomes. So, say for example, if we assume this to be the mom chromosome, the chromosome inherited from mom, this one to be inherited from the father, they have a pair of this, the same chromosome inherited, like two different variations of it, or two different versions of it. During meiosis, the homologous chromosomes separate, and then they further divide down the sister chromatids, forming gametes. Now, the number of genetically distinct gametes possible is 2n, and n basically represents the pair of homologous chromosomes that a parent has. Because humans have 23 pairs of chromosomes, the number of genetically distinct gametes possible in a human is over 8 million, which basically is the reason why there is so much genetic diversity and variations in humans. I hope you get this, and the next lesson we would kind of go over some questions to make sure that we understand all these terminologies. Thanks.